Good morning, you guys. Ms. Shapiro here. Hopefully you've seen a few of my other videos that I've made for you guys. And today I'm here to talk to you a little bit about the AP Environmental Science exam, specifically FRQs number 1 and number 2, although this video is going to focus mainly on number 1. If you're looking for information on number 2, there should be an entirely separate video for that one, so just go ahead and close the screen out and look for that one. I'm going to pull up my screen here and share it with you guys. My face should be minimized, but you should continue to see and hear me throughout the entire presentation, okay? I encourage you guys to pause as we're moving along and just kind of make sure that you fully understand things before you're moving on. Okay, so give me just a minute to set up my screen. And you should have a viewing guide available to you. I'm going to go ahead and open up my viewing guide so I can remind you guys where to record things, stuff like that. So if you need to pause this video for a few minutes and then just go ahead and make sure that you have that pulled up. We'll be ready to go. I'm also going to set a little bit of a timer, that's mainly for me. We don't want these videos to last too long, so this will kind of just give me a good idea about how long we've been started. So let's go ahead and start talking. The AP Environmental Science exam is composed of 3 FRQs. The first one is a FRQ that is solely about experimental design, and it's designing and evaluating investigations. So today we're gonna review the science practices that are necessary for the exam. We're gonna review the format for the exam and key components that go into FRQ number 1. And if we need to, we're going to review some content pieces that are important for your AP Environmental Science exam. I'm going to see if I can't just move this to the top. Okay. So moving right along, there are 7 science practices. So that's the first part of your viewing guide. What are the science practices? So I'll go ahead and give you a minute to stop and to record these. When you're ready, you're going to press play and you're going to hear me talk about each of them. So the science practices are hit throughout any portion of your exam, so multiple choice or free response as applicable. The first one is to explain concepts. So this just means you're going to explain to me how energy flows through a food web. You're going to explain to me how eutrophication works, something along those lines, okay? Science practice 2 is visual representation. So that means you're going to explain a model. So for example, if the demographic transition model was presented to you, you're going to be able to explain how countries move through that demographic transition model, how their economic status changes, how their birth rate changes, things like that. Another example of a visual representation might be, sorry, and I say a lot, I know, I'm trying really hard. A second example of a visual representation might be a graph, a figure that shows plate tectonics, and that might ask you some questions about plate tectonics, or a figure that shows you wind patterns, and you're going to have to decipher some information about winds. The third science practice is a text analysis. This is only going to be found in a multiple choice portion of the exam. So if for some reason your exam doesn't contain a multiple choice version, such as in 2020, you will not have any kind of text analysis. But this portion will ask you to read a text and will ask you questions about that text. Science practice 4 is about scientific experiments and investigating a design. This is hit heavily upon in FRQ number 1. So there are 4 parts to this, and we're going to go over those in just a minute, okay? There are 3 more science practices. Again, I would encourage you to pause this video and to record them. And science practice 5 is data analysis. This could show up in your FRQ number 1. This is going to be some type of data table or some type of graph that you're then going to have to interpret. You're going to pull information out. You're going to make claims about this data. Overall, we see that air pollution increased. Overall, we see that areas of dead zones increased. Overall, we saw that human population levels in Japan declined. So we're going to make some type of claim, some type of trend about some data, and then you're going to be provided evidence. That's numbers. So your claim and your evidence are very important in your FRQs, and we're going to see these are presented heavily in Science Practice 5, where they ask you to interpret and analyze data. Moving on, Science Practice 6 is mathematical routines. This could appear if there is an FRQ number 3 on your exam, although that's not the case in 2020. It will appear in all of the other exams as well as in multiple choice portions of your exam as well. Remember that there is a calculator allowed on the AP environmental exam, but if you don't know how to set up the problem, the calculator doesn't really do you a lot of good. And so we want to ensure that we're setting up problems accurately, that we're utilizing units, making sure that those units cancel, and we're calculating an accurate number and providing units in our answer. The last environmental, or the last APES science practice that you guys have to be familiar with is environmental solutions. So this means that if I present data with Science Practice 5, I give you, say, a graph that talks to me about air pollution levels. I want to know what are some solutions to those air pollution levels. Maybe things like catalytic converters on cars, an increase in mass public transportation, and an increase in rooftop gardens or other green spaces that try to purify and cleanse air. Maybe the graph is on habitat destruction over time, and I'm asking you to evaluate the causes of habitat destruction and then what are some possible solutions to habitat destruction. And you might tell me things about limiting population growth or vertical growth of populations. Maybe you would talk to me about a decline in ranching or farming that is often the cause of habitat destruction. So in this case, you're going to take the other science practices that are usually presented in some type of visual or some type of data analysis. They're going to present an environmental problem. They're going to ask you, what are all of the possible solutions to this problem? And then which solution is best? So take a look at 7.E and 7.F. We're going to come back to those in just a minute, but they're pretty important. So that was the first portion of your science, of your viewing guide. If we move on, specifically, FRQ number 1 is a design and investigation. So under what type of question is FRQ number 1? It's a design and environmental, or design and investigation. It's going to have 4 main components, and I'm just going to read them off the screen. You're going to describe a concept. So maybe they're going to give you some data on windbreaks and forestry. Okay, maybe forestry decline over time. So forests are going down. Maybe this is because humans are destroying the forest, or maybe it's because they're being cut down for a specific reason. So they're going to give you a scenario, either with or without data. And then I want you to explain the concept. Well, we see that habitat fragmentation is going on. We see that clear cutting is going on. If you don't know what clear cutting is, you're not going to be able to describe what concept is being described or being asked of you in your question. So just because they're giving you information doesn't mean you don't have to know your APES content. I would encourage you to maybe make a 1-pager note for each of the units. So go through and on a piece of white copy paper or computer paper, fill up one side, front and one side on the back of content about each of the units. What are some major trends that you see? What are some major vocabulary words? And define those vocabulary words. That way, when you're taking your exam in 2020, you can reference those. In other years, they're great study tactics, but your exam will be closed note. They may also ask you to analyze data or representation. So maybe they're going to give you a graph about how forests have declined over time, how forest cover has declined over time, and you're going to have to draw out trends from that particular data. Then they want you to analyze the research. So maybe they'll give you something like researchers went in and looked at forest decline over time, blah, 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 okay? How might we look at an investigation that tests regeneration of this forest? We took shelter wood cutting and selective cutting and strip cutting, and now we're going to design an investigation on which is the best type of sustainable forestry. Okay, they may move between topics. So maybe they'll start in forestry, but then they'll ask you some questions about the animals that are going to be impacted by clear cutting. And so you'll have to know about R selected species or K selected species. Okay, this is just an example, but they may start in a particular unit and then move through different units. So it's important that when you're reviewing, you understand how the different units and how the different concepts are related to one another. And then they're going to ask you again, what's the problem here and what's the possible solution? You have to pick from any number of solutions and tell them which one you think is best. Okay, so it was a pretty long-winded way to answer what type of investigation is FRQ number 1. But the main thing is that there's some type of investigation that they have done and they're asking you to evaluate, or they're asking you to set up. Okay, so let's move on further and look a little bit at some of these science principles. We talked a little bit about concept explanation. This is just saying what is going on. So for example, in our deforestation example, they might ask you, a forest has been cut down by clear cutting. What is clear cutting? Okay, and then you're going to have to explain a few things. Maybe they're going to give you a picture of different types of sustainable forestry, or they're going to give you types, or they're going to give you like a amount of forest that was cut down in each year. Okay, that might fall under a visual representation. Maybe they're going to show you strips of something or going to show you plots of specific trees. Typically, a visual representation is a map or like a food web, a food chain, a cycle, a population pyramid is a visual representation, along those lines. It's not generally a graph. Then they're going to ask you about a scientific experiment. So let's take a look more specifically here. They're going to ask you about a hypothesis, and so we'll go over what is a hypothesis. They're going to ask you about the method. Was this a valuable way to investigate this particular problem? They're going to ask you to make some observations or how you would collect data. And then they're going to ask you, how might you change this in the future? Or if you change this, what would happen to the results? The last component, again, is that data analysis. So pulling trends off of data, explaining patterns and trends, and explain what the data implies. Well, we see that over time, birth rate has fallen. This must mean that costs of raising a child increase. It must mean that women have access to education and family planning. And it must mean that the government is providing some type of social security so that families don't feel the need to have children in order to take care of them later on in their lives, okay? So what does the data tell us about environmental situations? And then the very last one, again, those solutions. So if we see that birth rate has fallen significantly below death rate, okay, our population is starting to shrink, what's a possible solution to that? Well, you might have some type of tax incentive that taxes people that don't have children and encourages people to have children. Maybe we're going to have a government program that offers increased housing or increased time off for individuals that have more children in that particular population, okay? So again, here we're looking at all of the possible solutions and then which solution is best. That goes back to 7.C, describe advantages, disadvantages of all of the potential solutions. Okay, there are four main big ideas in this course. The first one is energy transfer, and specifically we're talking about energy usually within ecosystems. Then we're talking about interactions in Earth systems. So this is relating things like winds and oceans and plate tectonics into biomes, into ecosystems, into how the biomes form with altitude or with latitude. So if those are not words that you are familiar with and you couldn't tell me, well, how do winds impact biomes? You need to pause this video. You need to find some resources on Earth systems, that's Unit 4, as well as biomes, that's Unit 1, and go back and kind of refresh your memory. And then we're going to take that a step further. How do biomes interact? How do species interact with their environments? So how does forest cover impact biodiversity? How does barrier islands help protect coastal ecosystem? Things along those lines. And then sustainability. So how are species, humans, impacting their environment? Well, we're withdrawing resources out. We're withdrawing oil, fossil fuels faster than they're being replenished. So we're going to find an alternative to fossil fuels. Or we're going to come up with a solution that combines fossil fuels like natural gas with a more sustainable solution like solar or wind. Okay. Don't be surprised if some of these FRQs incorporate all four of these big ideas or at least 2 or 3 of them. All right, so here is the part on your viewing guide. It says, in the box below, describe each of the following parts of an investigation or of an experiment. And so we see that we have to go over a couple of different parts. So an experimental design is a set of steps to yield valid results. So for example, we want to make sure that our results are repeatable. Okay, we want to make sure that if somebody else did the same investigation, they're going to get the same results as we are. If it's not repeatable, it's not a very good experimental design. We want to eliminate errors, and we want to make sure that our investigation is due to the factor that we're testing, not something else. So for example, if you were growing a bunch of plants and you're going to grow plants in light and you're going to grow plants in different colors of lights, and then you're going to grow some plants in the dark, you want to make sure that your plants aren't growing because you gave one of them a lot of fertilizer and you didn't get any of them, and you didn't give the rest fertilizer. Or maybe it's the fact that we want to make sure that our plants are growing due to the light and not due to some other factors, such as the presence of a hormone or the presence of fertilizer, something along those lines. Okay, and then all experiments are based on a testable, repeatable hypothesis. A hypothesis is not just like a guess, but it's something based on research that we can say. So for example, if I change this, if I put plants in a variety of different light conditions, then the one that's placed in natural outdoor sunlight is going to grow the most, and the one placed in the dark is going to grow the least. So that's a hypothesis. It's going to tell us what we think is going to happen based on research or based on prior knowledge. We're going to come back to the hypothesis, so if you want to skip that very first box, we might make a few notes in it later, okay? So the IV stands for independent variable. The independent variable is also called the manipulated variable. This is the thing that I change. I for independent variable. I am the researcher. I am going to change it. Have I said I enough? Okay, I change it, and so we call it the independent variable. And this usually has levels, meaning that I'm going to change it in small increments. I'm going to put a small brine shrimp, which is kind of like a little sea monkey. I'm going to put it in different amounts of caffeine, in no caffeine, in 5 milligrams, 10 milligrams, 15 milligrams, and 20 milligrams. And I'm going to see how much that brine shrimp starts to move and freak out, okay? And so those are my independent variable levels. I'm changing if it's exposed to caffeine or not. The levels are which I'm changing are 0, 5, 10, 15, and 20. Independent variables should have at least 5 levels. So if their AP exam asks you to identify levels of an independent variable or identify the independent variable, you're going to want to come up with at least 5. Okay. Other examples might be plants grown in dark sunlight, red light, blue light, and green light, okay, or purple light. Those would be my 5 levels of my independent variable. Okay, and it's independent because it's what I'm choosing to change. The dependent variable, or the responding variable, is what responds when I change my independent variable. So it depends on the independent variable. It is the dependent variable. So in my example of brine shrimp, it would be how much does the brine shrimp move when exposed to caffeine? I know you guys are all out there drinking your Monsters, drinking your Rock Stars, and then you start to get the jitters, or maybe you get really, really hyper. Okay, so I exposed you to caffeine, you have increased energy levels. When I expose that brine shrimp to caffeine, how much does it move? That's my dependent variable. If we go back to my plant example, how much does the plant grow? Okay, so does it grow none? Does it grow 1 foot, maybe 10 inches, maybe 1.5 feet, maybe 20 inches? Okay, so my dependent variable depends on the independent variable, and the independent variable is what I am changing. You don't need levels of your dependent variable. It's just kind of the response. The next thing we could talk about is the control. Okay, and that's a portion of the independent variable. The control is the level of the independent variable that's most like normal. And it's what we want to compare everything to. So brine shrimp, I know you're going to be shocked by this. Brine shrimp do not normally live in caffeine. So we want to compare it to a brine shrimp that wasn't exposed to caffeine. We want to compare our plants to the plant that was grown in normal outdoor sunlight, because that's the plant light that most plants are exposed to. Other examples of control might be if you're looking at how many people get sick If they're exposed to something, you're going to look at somebody that wasn't exposed. Another example might be how do plants grow in response to salt? Your control is a plant grown without salt. So it's normal conditions. So one level of your independent variable always has to be your control, or the thing that's most like normal conditions. Sometimes we'll have a negative control. So this is something in which it wasn't exposed to anything. So for example, a plant grown in no light is typically a negative control. And then grown in sunlight is our regular control. And then grown in blue, green, purple, red light would be levels of an independent variable. So if you see something about a negative control, it's just removing everything, so it makes sure that we're testing what we're actually wanting to, or the results are from what we actually want to test. Okay, moving right along. So let's say you're a farmer and you want to test your crop yields. You want to test how much your plants are giving off. What would be your independent variable and your dependent variable? So I'm going to give you a moment to pause this video, write in your box, what's the independent variable, what's the dependent variable, then press play when you're ready to hear the answers. Okay, so my level of independent variables would be crops grown with pesticides and fertilizer. That's what I'm changing, exposure to pesticides and fertilizers. And then my dependent variable would be how much the crop yield either increases or decreases. Usually our hypothesis is given in an if-then statement. So if the independent variable, then the dependent variable. If I put brine shrimp in different amounts of caffeine, then the one that's placed in 20 milligrams of caffeine will move the most. If I put plants in various amounts of light, the plants in red and blue light will grow the most because that's the wavelength that plants will absorb. If I put pesticides and fertilizers on my crops, then crop yields will increase compared to plants that did not receive any pesticides and fertilizers. Okay. Other things that you might need to consider would be things like duration. How long are you going to let your experiment last? Are you going to put your brine shrimp in for 5 minutes or 5 hours? If you put them in a rock, like a what do you call those? A monster or a rock star that has a ton of caffeine and a ton of sugar, and you leave it in there for 20 hours, your brine shrimp's probably going to be dead. Okay, so we want to think about how long are we letting things go. In trials, how many times are we going to repeat it? So for example, I'm going to let my plants grow for a 2-month period, and I'm going to do 5 different plots where I repeat the same thing. That's a trial. Okay, I'm going to let my plants grow for 6 days, and I'm going to put 5 plants in each level of independent variable. That is a trial, okay? And so each one of those plants produces a data point. If it just is done one time, that's typically not enough to give us valid results. And remember that we want our experiment to be valid. Constants are things that we hold the same. So for example, where we got our brine shrimp, we don't want super healthy brine shrimp in one experiment or in one level, and then like sickly, already dying brine shrimp in another one. So the health of our brine shrimp would be a constant. Maybe if we're talking about our other experiment, it's the type of plant. You don't want to grow radishes that grow really, really fast against a very slow growing plant like an orchid. Okay, those things are not comparable. So constants are things that we hold the same so that we can do a comparison. So how much water are your plants given? How much pesticide are they given? How much fertilizer are they given? These are all constants that we would want to be considerate of. They're all grown in the same type of soil. It's always the same type of plant. Maybe we're going to grow in 5 different plots, and we're going to expose each plot to different levels. We're going to have 5 rows in each of our 5 plots, and each row is going to receive the same amount of fertilizer. Okay, that would be a constant, and that would describe trials. Okay, so I'll give you a minute. I'm just double checking our viewing guide here to make sure that your examples are filled out and that your viewing guide is filled out. So I'll give you a moment here to pause the video to catch up, and then press play when you're ready for me to keep going. Okay, so here we go. This is just a filled out version, so make sure that yours matches mine. I'll give you a minute to pause and to catch up. Okay, so here's an example of how it's going to look on the exam. So I'm going to give you a minute to just go ahead and pause, read over this, but I'll give you a brief synopsis. Basically, a student is growing two species, species A and species B. In group 1, they're grown individually, so they're grown in separate containers. In group 2, they're grown together. And I don't know on my recording if my face is in the way, so I might just move this kind of out of the way here. And so I'll give you a moment to look overall at the data. So again, we have two species, A and B. In group 1, they're grown separately. We see that this is the data for A, this is the data for B. In group 2, they're grown together. Okay, here's the data for A, here's the data for B. So then on our exam, it's going to ask us, and I want you guys to continue to see my shiny face. I put makeup on for you today, so I want you to see it. I'm going to give you three components, 3 parts, okay? So the first one says, as shown in the table, the student established treatment group 2 with 5 individuals of each species. Provide reasoning for the reduced initial population sizes. So here in group 1, we started with 10. In group 2, we started with 5. How come we went down by 5? Okay, so I'll give you a minute to think that over and maybe record somewhere on your sheet. Number 2 says the student claims that A and B compete for the food source. Provide 2 pieces of evidence from the data that support that claim. Evidence means numbers. So if it asks you for evidence, you're going to want to cite numbers. Well, species A and B must compete for the same food source because when grown alone, A reached 1,500, and when grown alone, B reached 700, but when grown together, neither one of them reached the same level of individuals. So you're going to want to reference numbers, okay? So evidence must have numbers. And then number 3 asks us to predict 2 features that will most likely limit the population growth. So here we've shifted. We've moved from talking about an experimental design, now we're talking about content. What's going to limit the population from growing? Okay, so I'll give you a minute to pause, make sure you have some answers, and then press play when you're ready for me to go over these answers. Okay. So here are our results. We see that reduced initial population sizes keep the total of number of organisms the same in the container. So if we go back, we started with 10. Here, because they're grown together, 5 plus 5 is 10. And so we're going to say we want to keep the same number of individuals at the same time, and it serves as a control. Number 2 asks us for 2 pieces of evidence. And there's any number. When we're comparing A to A, the growth rate is faster in 1, and it's slower in 2. They must compete for the same food source, so because they're competing, they're not growing as fast. Maybe we see that A, you saw 1 went to 1,500 and 1 went to 1,400 in 2, and so because they're not as highly populated, it must be because they're running out of food. If you're comparing B to B, the same thing, they do not grow as fast. And A, they do not grow as high. In fact, 2 goes all the way to 0, so they must not have enough food, they're dying. Okay, and then 2 factors that are going to limit a population growth, food, space, access to dissolved oxygen, because I'm pretty sure this is aquatic, and then waste buildup. As toxins and waste build up, things are going to start to die. Remember, they're in a container. You didn't have to include all of these, just 2. Remember that these are density independent factors, or density dependent factors. So if you don't know what a density dependent factor is, go back and look that up. But that's something that we need to know about things that impact populations. These are both density dependent controls. Okay, so remember to be concise and to the point on your FRQs. Only give for the things it asks for. Ooh, preposition at the end of a sentence. If it asks for 2, give 2. Do not give 3. They will read the 3rd one, but they will not award you any points. Don't assume that we know what you mean. If you're thinking it, you should put it down on that paper. I cannot grade what I think you're saying. I have to grade what's on the paper. So do not put down something and be like, oh, she'll know what I'm talking about. I absolutely will not know what you're talking about, okay? So be concise, but be thorough. Do not feel the need that you need to keep writing if you've already said stuff, but if you can ask yourself why, and you haven't put that down on your paper, then you need to keep writing. Okay, I have listed out the task verbs. We're going to go over these just kind of briefly. We have been talking for about 27 minutes, and I want to make sure that we have time to get to some examples. So these task verbs, so describe is provide characteristics. Describe typically means make some type of statement or definition and give an example. That will almost always earn you the describe points. Explain is a little bit more. Explain means make connections. So A causes B to happen, B causes C to happen, and C and A are related in this way. You have to go back. So A to B, B to C, C to A. You have to make connections. Typically, we want you to explain how this occurs or why this occurs. Okay, why does this the best outcome? How did this happen? What were the causes behind all of this? So explain, you're probably going to be writing the most. Identify, you're going to be writing the least. Okay, one sentence. Okay, it's going to ask you to identify the primary producer of a food web. The primary producer of a food web is blank. It needs to be in a complete thought, but we don't care so much about capitalization or anything like that, but we are looking for a complete thought or a complete sentence. If you just write one word, you may not get full credit. Justify is like defend yourself. Make an argument and defend your stance, okay? Make a claim is kind of like identify. It's just one statement. The data increased. It increased from 10 to 15, whatever it is, but be specific, but be concise. And then propose a solution. This is, how could you fix this? And why is this the best solution that you could come up with? So if the problem is deforestation, then the best solution would be to limit population growth. That's going to keep our forests intact because people aren't expanding out into new spaces. This is the best one because it stops people from moving into new areas and allows the habitat to be maintained. Okay, so at this point, I would like you to encourage you to pause, maybe take a little break, go outside for a few minutes, but also record any questions that you still have that you may submit to your teacher. We're going to move on into some examples. Let me just double check that we're right where we need to be. I have 3 examples after this, so we are right exactly where we need to be. Okay, so here's our first example. This one is about air pollution. If you're looking at the viewing guide, I did link the article that I got this one from, so you can go back and look at the data that contributed to this. So in this example, scientists sought to collect data on the association between air pollution exposure and dementia. Using data from Italy, researchers measured levels of tropospheric ozone, particulate matter, and NOx, NO2, or NO, across various parts of Italy. Then they correlated these levels with participants' addresses. So if you're taking this exam in 2020 and you don't know anything about tropospheric ozone, you need to go back and review, okay? Everybody needs to know some stuff about tropospheric ozone because air pollution is fair game for your test. So please just make sure that you have gone back and you've reviewed the causes of tropospheric ozone. Remember that ozone is good up high, stratospheric, bad down low, bad in the troposphere. Ozone is good. It helps pale pasty people like me not get sunburned, but it's really only good in that stratosphere. Otherwise, it's a air pollutant. It causes respiratory illness in the troposphere. Remember, that's the lower part of the atmosphere. So again, if you don't understand the content, those things aren't going to mean anything to you. You're going to have trouble answering the question. Then they looked at various hospital records of the participants who complained of or had symptoms of dementia. Remember that dementia is kind of like Alzheimer's is a type of dementia where you're losing your memory. I want to know what's the independent variable, the dependent variable, control, constants, and a hypothesis. So at this point, I'm going to ask you to pause your video. I'm going to ask you to record your answers in your viewing guide. When you're ready for me to go over the answers, you'll just hit play. Okay, so here are some of our results. So the independent variable is whether the participants were exposed to air pollution. I didn't really manipulate it, but it was the thing that participants were looking at different parts of, okay? Obviously, we're not gonna go and expose people to air pollution. So this was like a natural study. So what was the thing they changed? if they were exposed to air pollution. If you wrote down something about addresses or comparing the different people's addresses and therefore what they were exposed to, that probably would also be accepted as well. The dependent variable is what depended if the people had dementia or if they were complaining of dementia symptoms. And then we went on to talk a little bit about the control, okay? The control is dementia in an area that didn't have any pollution. So we're gonna find data from outside Italy, I think this was, yeah. So maybe we're gonna compare it to data in rural United States that typically has less exposure to air pollutants. So we're gonna compare it to rural Italy. Actually, rural Italy is probably better than the rural United States because there's some other constants that we want to control. Maybe they're going to compare it to other people in the Mediterranean region who have a similar diet, something like that, okay? So the control is the dementia in less populated areas. It's what we're going to compare our data to. Constants would be only looking at Italians or only looking at Mediterraneans, right? We're not going to compare them to Americans because Americans have a different diet. We have different exercise. We live in less densely populated areas, so that's going to all control the things. We're going to look at the same age range, right? We don't want to compare rates of dementia among people that are 70 to 80 to among people that are 10 to 20. Those things are not going to be the same. So we had to compare it to the same people that are in the same age range. And then how they took the data for dementia. So, right, was it just like, oh, I forgot something yesterday? Or was it like, I'm always forgetting something over long periods of time? Okay. So our hypothesis is something along the lines of if individuals are exposed to various levels of pollutants, then they are more likely to suffer from dementia. An even better hypothesis, now that I'm thinking about it, is if individuals are exposed to high levels of air pollutants, so not just various levels, but high levels of air pollutants, then they are more likely to suffer from dementia. Alternatively, you could say the opposite. If individuals are not exposed to air pollutants, they are less likely to get dementia. Okay, so the inverse is almost always true. So I'll give you a minute to pause, make sure you've made any edits, and record any notes that you may need to from this example. Example number 2 is about deforestation and sediment flow. And so again, I've linked that there for you. So this one says, when land is initially cleared for agriculture, soil erosion can occur. So remember that agriculture is the leading cause of deforestation in parts of the Amazon. Usually they're raising crops to give to cattle, or they're raising cattle themselves. So the best way to stop deforestation is to limit the population growth, because we're trying to feed all these humans, or maybe change what the population is eating, less beef consumption. I'm not saying you have to become a vegan or a vegetarian, because ain't nobody got time for that. I'm saying less, okay? Or buying locally raised meat as compared to imported meat. When agricultural practices such as tilling, remember that tilling is when we mix up the dirt, and farmers till every spring as they get ready to plant. So when agricultural practices such as tilling occurs, that soil erosion can increase. Remember that erosion is when wind or water takes soil away from its initial location. A city planner wants to investigate the erosion that occurs when various human disturbances, such as clearing land for agriculture, occur near a river. So what's the independent variable, dependent variable, control, constant, and hypothesis? I'm going to give you a moment to pause, record this in your answer sheet, press play when you're ready for me to move on. Okay, so the independent, oh, I'm sorry. I think I have some errors here. Hold on, let me make some edits and we can go back. Okay, I had to do some updating. Sorry, guys. So here's the correct answers. So what's the independent variable? So the independent variable is the type of disturbance. So our scenario tells us that various human disturbances, such as clearing for agriculture. So maybe we're clearing by slash and burn. Maybe we're clearing by conservation or conventional tillage. So we're just mixing things up. Maybe we're not clearing at all. We're doing some type of conservation tillage. Or maybe we're clearing versus like some type of contour farming. Okay, so our independent variable is what I changed. Maybe we're doing clear cutting. Maybe we are not clearing at all, okay? Our dependent variable is the amount of erosion that's going to occur, so the amount of soil that's going to end up in the nearby river. Our control is an area that wasn't exposed to the disturbance at all. And our constant is maybe how long after this disturbance did we measure. It's not fair to say, okay, well, we cleared yesterday and we're going to measure the amount of soil, and somebody else is going to be like, okay, well, we cleared 3 weeks ago and we're going to measure the soil. So we're going to think about time, we're going to think about duration. Okay, I'm going to measure at 1 day, 1 week, 2 weeks, 3 weeks, 4 weeks. Okay, so those are constants, but those are duration that we're going to think about in our trial. It's not fair if some people clear just like 1 square foot and some other people clear like 100 square feet. So how much land are you going to clear? Okay. How much are you going to clear? Are you going to clear in multiple plots? So we have some type of trial going on. Okay. So it's all some things to think about. This isn't all inclusive. There are other things that are probably acceptable. You can check with your teacher. And there are probably some things that we've left off here. Our hypothesis says that if slash and burn technique is used to clear land near a river, then soil erosion will be its highest. There are other acceptable answers. If you put down a correct answer on the AP exam, you will be awarded points, okay? But we tend to see some of our, don't go like off the beaten path. Don't come up with like a random answer that you know is correct, but not kind of mainstream. Stick to basic things, stick to things that you know, stick to things that you've talked about lots of times, okay? If it's factually correct, we're going to give you points, but it helps the reader, it helps us, the people that are grading it, if they're not like off the wall things that we have to go and look up. I just want to make sure we're doing okay on time here. Okay, we have our last example. Pharmaceuticals in the water. So this one says that, it says researchers in parts of Asia recently conducted water tests at various locations on nearby water sources. Their tests indicate that pharmaceutical drugs, including antibiotics, pesticides, birth control, and drugs used to treat heart disease, have all been found. Concerns of increased concentration and the impact that it might have on nearby biodiversity, as well as people who consume the water, have all been raised. Researchers are worried about the impact that contaminated water might have. Go ahead and pause your video, complete your questions, and then go ahead and press play when you're ready. Okay, so here we go. Our independent variable is where we are testing, okay? And so it says they are going to conduct water tests at various locations on nearby water sources. So where are we testing is the thing that I'm changing. If I only test in one spot, then I don't really have an independent variable. They've already told you that pharmaceuticals have been found. So now we don't want to know if they're present, we want to know where they're present. And so that's my independent variable. The dependent variable is the type of contaminant found, an antibiotic, pesticide, etc., as well as the concentration. So is that antibiotic found in high amounts, maybe 100 parts per million, maybe 1 to 5 parts per million. So the concentration as well as what type. A control would be the concentrations in uncontaminated drinking water. A constant would be like, when did you collect it, the time of day, the amount that you collected. If you collected 100 liters versus only 1 liter, then obviously those concentrations are going to be different. A consistent location for collecting. So it's not okay if one time you collect on one side of the river and on the other time you collect on the other side of the river. So we want to always test in the same spot. We want to test a variety of spots. But we want to always test from the same spot. And then how we collected it. We don't want one person to maybe contaminate theirs while other people do not. Okay, so those are all constants. And then our hypothesis would be if various water sources are tested for pharmaceuticals, then the levels of contaminants can be determined and evaluated. Okay, so hopefully this helps clear things up. If I were to take this last example, I might ask you some questions, right? Some solutions. How would you fix this? How would you change your water purification system? If you're taking this in 2020, that's not content that you have to know, but if you're taking it in any other year, absolutely. So how might you fix this problem? What's the environmental solution to contaminated water sources? You might talk to me about bioaccumulation, biomagnification. You might talk to me a little bit about how they got there with surface water runoff. So be able to talk to me about this. What would be your next steps? Well, I would want to check to see how much contaminants were found in drinking water. I would check to see how much is being excreted by the body, so we can maybe do some type of bioaccumulation study, things along those lines. So we want to make sure that we are extending this into future investigations, because that's going to be a good place for them to ask you a couple of questions. Okay, so just as a review, we want to talk about the independent variable. That's the thing that I change or I manipulate. We want to talk about the dependent variable. That's the thing that changes as a result. We want to talk about controls, things that we're going to compare them to, not a constant, but a control. What are you going to compare it to? And then we want to talk a little bit about how long, trials, those kind of things. Okay, but if you know independent variable, dependent variable, and control, you're probably in a great spot. At this point, I'm going to ask you, how comfortable do you feel with this? Do you think you need to go back and rewatch portions of this? I can talk really fast at times, and I know that. I tried to slow down, but remember that you can always pause and go back to rewatch. Okay, what are your next steps? How should you move? Okay, what should you do to move on? Things like that. And what else should you do in order to review? Okay, I hope that you have found this to be beneficial. If you have any questions, please reach out to your teacher, and she'll be able to, or he or she will be able to help you. I wish you guys the best of luck on your AP exam. I know you're going to do great.